everyone. I'm Yume Imada, an assistant professor at the Ehime University. I'm so grateful to Akira for inviting me to this lecture. It is my pleasure to speak here today. I entitled my talk as The Web of Life on Bryophytes carpets. Since I started my career as an ecologist, I have studied the associations between bryophytes and animals, especially insects that feed on bryophytes. So I'm going to talk about it. So first, you need to know what the bryophytes are. The bryophytes are the earliest branches of extant land plants, which originated in the mid-Ordovician. That is almost at the same time as the insects had emerged. And they comprise three divisions: liverworts, wormworts, and mosses. The relationships among these three groups are not well understood, but recently many studies suggested that the bryophytes are monophyletic, so they evolved from a common ancestor. These groups are evolutionarily distantly related, and each differs in form. Well, mosses are among the most species rich, and they are also morphologically diverse. One of the common features among these three groups is a life history. I mean, the alternation of generations. The gametophytes are the dominant life stage, and so the leafy organs, leaves and stems are all gametophytes. And the sporophytes only develop in a very short period of time, and they parasitize on the gametophytes. There are some notable morphological, physiological, and ecological features that bryophytes are so distinct. First, the bryophytes do not have vascular tissues. The picture on the left is the cross-section of the moss stem, and you cannot find a central bundle in it. They are also unique in water transpiration system. They lack the ability to maintain and regulate water content for a long time, but they quickly acquire water from the environment. This feature is called as poikilohydry. This makes them flexible in water availability, and because of that, some species have a wide range of potential habitats. They exploit arid small space, like at the corner of the pavement, like that. Although most species prefer much more humid environments. They do not have roots, and so they do not need to anchor themselves in substrates. They do actually grow in a range of substrates. Sometimes we can find a statue of Jizo like that. Is wearing mosses like he's wearing blanket. And we Japanese people appreciate it as a symbol of eternity. I'd like to show you how the bryophytes carpets look like. Now we see the forest floor along stream banks. It's decorated by the patchworks green to yellow, and each of them is different mosses. Each species has a preference for the microhabitats, depending on the levels of moisture and access to the sunlight. The structure of patches are largely affected by the types of growth forms, such as tall tufts and ferns, many other types. As a part of the ecological functions of the bryophytes, they can be home for diverse microarthropods, which usually live in or near the soil. Bryophytes can retain humidity and protection against sunlight and predators. And so many tiny arthropods like springtails, mites, and nematodes are commonly found on moss patches. Some species even feed on bryophytes. But I must say there are very few reports on the animals that they use bryophytes as foods. I suspect that it's only under investigated because bryophytes are tiny and what's in there are even smaller. Still, many researchers believe that bryophytes are not available foods for many animals in terms of nutritional values. The bryophytes are also chemically dependent, which make them so distasteful. So I have been to mossy forests wherever I could go and have studied life histories of some interesting insects that cannot live without bryophytes. The bryophytes are usually found in a humid forest and places near rivers and streams but they really dominate in that landscape. So I usually look for some clutters of bryophytes and stream bunks and carefully look for the tiny animals in the patches. But this forest is a pristine forest in Nagano Prefecture in Japan. Only the coniferous trees can grow in such subalpine zone. In the course of my field work, I have discovered some new interactions between bryophytes and insects, which were previously unknown. Most of them were found in the temperate forests in Japan or in North America, but you may find something in the tropical zone forest. And a very important discovery was actually made in Peruvian lowland rainforest in 2015, which is already published in 2018. As the subtitle shows, this is a discovery of a new interaction known as galls. The insect galls are common among angiosperms, but they had not been known until we found that in Peru. This locality is a waterfall, and we examined this liverwort species Monocolia gotzi, which was abundant there, and some of them had a hemispheric swelling on the dorsal side as shown with the arrowhead. It turns out that this structure is induced by the insect living inside the plant. We made some thin cross sections of the plant tissue, and then as you see, there was a vacant space without plant cells inside the gall. This space is technically called as larval chamber, where the insect larva feeds on the plant tissue and develop in it. You see a nice piece of fecal pellets, which is defecated by the larva, and the pupal exuvia. So it means that the larva became pupa and it escaped from the room. This sort of gall structure is induced by the insect in the gall. The mechanism in general is not well known, but basically it is thought that the insect secretes a sort of plant hormone and it causes the abnormal development of the plant cells. And what's important about the liverwort gall is that the forms of the cells surrounding the larval chamber. You may see that the cells have 3 layers with different sizes and forms. The cells in the inner layer consist of large and rounded cells, and the middle layer is kind of flattened and particularly abnormal. So such differentiation was not found outside the galls. And this layer structure of the gall is interpreted as a way of response of liverworts to the insect gall inducer. So this is the first insect-induced bryophyte galls ever known, and no one hasn't found any other examples yet. So I'd like to move on to the main part of my talk. Well, I will introduce a piece of my work published in 2021. This is a miraculous case of an insect evolution, which took place on the bryophytes carpets. The insect larva shown here is Lioguma mikado, which belongs to the smallest family of crane flies, Slendrotomidae. What's surprising about larva is how it makes it blend well into mosses with complex traits. The larva has a body color and pattern of dark pigmentation, but a moist and translucent texture. What's more, you see many elongated structures which surround its body. The form of the larva is interpreted as an excellent example of camouflage. Hereafter, I call this phenomenon as moss mimesis. Here's a general evolutionary scenario of camouflage. Camouflage often evolves as protection against keen-sighted predators. In the evolutionary process of mimicry, the relationships between organisms at three different trophic levels are involved. They are predators, the primary driver, the model, plants in this case. Visual camouflage is often attained by combining some morphological and behavioral traits. Historically, many studied cases of mimicry have focused on the roles of color and pattern. The physical structures also play a crucial role in some cases, but they are rarely studied. There are only one famous example of such: narrow tails of some butterflies mimicking antennae of their hind wings, which acts by deflecting birds' attention. In this regard, the Cynindrotomidae can serve as a unique system for studying function of mimicry-related structure. This study is driven by two questions. First, I explored how their series of traits related to mimicry can be associated with the ecological background. And so I wanted to figure out their habitats and biological interactions with predators and host plants, which seem to be associated with the highly cryptic form of the Cynindrotomids. The second question refers to the function of the elongated cuticular lobes. This is a question from an ecomorphological point of view. So I investigated the host plants from which the larvae were collected. It turned out that some species only feed on some specific groups of mosses. 3 species listed here feed on Moniaceae in the field, particularly those shown in orange: Plasmodium, Rhizomonium, and Monium. These mosses are Acrocarps mosses, which is a type of morphological feature. The shoots of the Acrocarps mosses are usually unbranched, and importantly, they make erect tufts like small trees. Neugma nodicornis, that's a species of terrestrial moss-feeding Cynindrotomids in North America. This species even live in urban parks. So if we want to find the larvae of the species, Moniaceae moss patches are the place to look for. Well, there's a tip for finding the larvae in the field. You need to know the host plants and the feeding damages inflicted by the young larvae. The white spots that you can see are the cells devoid of chlorophyll. This is a sign of piercing and sucking feeding by the first instar larvae. The feeding marks are conspicuous on Moniaceae mosses, and yet, unfortunately, the feeding marks are not evident on the other groups of mosses. So if you go to marshy woodlands and see these transparent cells in Moniaceae, the chances are that they are under your feet. Some other species feed on hypnels and others, which are distantly related to Moniaceae. So these species tend to be generalists that accept many different groups of mosses. The species in hypnels are all Plerocarps mosses, which usually make fern-like appearances. They tend to form spreading carpets on various substrates like rocks and tree trunks or whatever. The host plant morphology differs between hypnels and bryales, that includes Moniaceae, and that should affect the appearance of moss patches, which would also affect the visual background of the insects. And I have observed the oviposition of adults to see whether they specifically lay their eggs onto the larval host plants. I examined 3 species, and as a result, they lay their eggs on plants. The oviposition substrates differ among species, but they do not always lay eggs on their larval host plants. Sometimes they do oviposit on mosses, but sometimes they do not. To reveal the evolution of cryptic mimicry in Cynindrotomidae, we are on the way to investigating the interactions with predators and host plants they are part of, and infer the trait evolution built on the phylogenetic framework. The result of phylogenetic analysis so far suggests that the terrestrial moss feeders can be clustered by the range of the host plants. Specifically, clade A is composed of the species feeding on Mooniaceae, and this clade is derived from the other groups of the genera, namely Hypnaeal feeders. I was able to collect all of the described genera of Cylindrotomins and compare the larval morphology, as I will later discuss. The terrestrial moss feeders can be characterized by having the dorsal lobes with complex structures with teeth-like forms. Other than the terrestrial moss feeders, two other genera are biologically distinct. The aquatic moss feeders tend to have long and thin cuticular lobes, whereas the terrestrial herb feeders have flat bodies and the lobes are inconspicuous. I remember the moment that I found the larvae of Phalacroxera tipulina at Mountain Lake in Virginia, that's in the United States. They occur at high density in a small pool of sphagnum bog. The larvae remain motionless on shoots of sphagnum mosses, but occasionally surface. They feed on a range of aquatic mosses, including Sphagnaceae and Fontinaceae. It's quiz time. How many larvae can you find on the leaf? Well, this is a leaf of Hookie. I wait just some seconds. Can you find one? Well, here's the answer. They are the larvae of Cylindrotoma, which usually stay on the surface of the leaves like that. They can be locally so abundant like this with several larvae on one leaf. Cylindrotoma represents terrestrial herb feeders. They have a vast repertoire of host plants encompassing 15 families of herbaceous plants. They show some behavioral features that are not in the other species of Cylindrotomins. They are susceptible to the motion of surrounding objects, and they show defensive behavior when threatened. This behavior may be interpreted as defense against predators or parasitoids. The larvae live on a flat leaf surface, a substrate, which provides no supporting structures to hold on to, and so they ensure their attachment via the suction cup principle and sticky gland secretions. The photo shows a larva is repeatedly stomping their thoracic segment against the plastic seat. The suction cup mechanism also has a role in locomotion. The larvae move around by inching when they transit from one leaf to another, and they fix themselves with their caudal end and straighten up sharply and then move forwards. The forebody often accompanied with a slow back and forth motion. Next, we will see what the fundamental structures relevant to crypsis are. Moss mimesis is achieved by a set of characters. The matured larvae exhibit two different body coloration components, which are the colored hemolymph, causing green or brown hues. The second one is the dark pigmentation on the dorsal and tegmental surface. The occurrence of the pigmentation shows a well-organized species-specific patterning consistent with each body segment. And the third element is, of course, the cuticular lobes. In some species, the color of hemolymph is polymorphic, either green or brown. But some other species do not have much variation in color. For example, Diogma brevipecten stay green and do not change its body color, whereas Triogma guanai only has a brown mold. The range of colors of each species seem to be correlated with the difference of the microhabitat. For example, Triogma guanai are not normally found in moss patches, but they are hidden in small crevices. However, the mechanism that determines the color is unclear. Finer instar larvae are most easily characterized by their elongated cuticular lobes, which are integmental outgrowths. A set of several lobes of various shapes are regularly arranged on the thoracic and abdominal segments and occur on the dorsal, lateral, and ventral sides. They vary in size, number, form, and ultrastructure across species. This figure illustrates the relative position and size of the lobes on the group of body segments from the prothorax to the anal segment. This figure illustrates, you can see that the configuration pattern is various across the lobes on the dorsal, lateral, and ventral sides. Let's take a closer look at the dorsal lobes, which shows the broadest range of variations and forms. And the dorsal lobes can be most detectable from potential predators. The dorsal lobes are particularly well developed and markedly differentiated among species on the abdominal segments. They are so short and tuberculate in terrestrial herb feeders, Cynodrotoma. The terrestrial moss-feeding species exhibit the most elaborate patterns, having teeth-like appendages, axillary outgrowths at the frontal side as yellow arrowheads. The dorsal lobes are extremely long and filiform, thread-like in aquatic species of Paracrocera. Although the functions of axillary outgrowth is so far unknown, they may help breaking up the caterpillar-like outline and facilitate the disruptive coloration in collaboration with color and pattern. The larval morphological features can be related to the preference for the host plants because the host plants serve as a visual background and thereby directly influence the success of camouflage. It's actually way more challenging to spot larvae in the moss carpets of hypnels, like in the above photos than the one at the bottom. So I suspect the species feeding on hypnels are visually more defended than the larvae on the erect tufts of Moniaceae mosses. Well, presently we are conducting quantitative analysis on how the feeding choices of host plants is correlated with the visual matching of the insects and the host plants in the framework of bird color vision. But still, the morphology of Cynodrotoma is curious, and so I dug deeper on the function of the lobes. The central question here is how a device for mimicry can evolve simultaneously alongside physiological, mechanical, and behavioral functions. The cuticular lobes have been hypothesized to serve as a tracheal gills in aquatic species, or an osmoregulatory organ, or a locomotion device in both terrestrial and aquatic species. Well, some previous authors pointed out the possibility that the lobes in aquatic species of Paracrocera are thin and long, and they resemble the gills of fully aquatic species like a Centropini moth. First, I consider whether the cuticular lobes can serve as a device for respiration or not, in the light of three standard methods for gas exchange in insects. The tracheal system connected to the spiracles, tracheal gills, and cuticular surface, including plastrons, which are water repellent structures that allow insects to use the air-filled tracheal system underwater. In general, terrestrial species of tipulits, crane flies, have a pair of posterior spiracles on the anal segment. But the structure of spiracles in crane fly larvae tend to differ between the habitat types, like aquatic versus terrestrial. And spiracles can even be non-functional in some aquatic tipulits. So I examined the spiracles for three species using the scanning electron microscope. The SEM images of both terrestrial Cynodrotoma and Eugma and aquatic Paracrocera species show that all examined species maintain the posterior spiracles, opening at the central scar plug and air piles. The important fact is that the spiracles of Paracrostotubulina are consistent with the idea that they are functional. In Paracrostotubulina, the spiracles lie in the retractable spiracular field and are surrounded by a hair fringe, which is generally thought to prevent water from getting into spiracles. Then I examined the ultrastructure of the integument. These images show that the cuticular surface of the Paracrostera does not bear bristles or hairs, which function for retaining air. The integument lacks potential aerial chamber, which is a three-dimensional meshwork structure with minute pores internally connected to the tracheal system. The photo in the middle is the cross-section of the lobe. And it shows that cuticle was too thick to let the air pass through. Interestingly, some characteristic ultrastructure of the integument, such as micropapillae, are hard to interpret. They're not comparable to any orthodox plastron structures known in arthropods. So overall, I could find no evidence for cutaneous respiration with plastrons in aquatic species. Another functional hypothesis is locomotion and attachment. Some previous authors emphasized the roles of the ventral lobes in the anal segment, as they are used for attaching the body to plant substrates. For example, Hinton, 1955, found that the ventral lobes of Cylindrotomys lack intrinsic muscles and thereby concluded that they are not prolix. But the internal structure of the dorsal and lateral lobes were not mentioned in previous studies. So I investigated these hypotheses in two ways. On one hand, the internal structure was investigated by making serious sections. On the other hand, attachment structure was examined by the SEM observation. To make a long story short, the ventral lobes did not provide much attachment device, and the results are not entirely novel. So I'm going to talk about the internal structure. I made a series of cross sections of larvae from thoracic to abdominal segments. I noticed that lateral lobes are different from the dorsal and ventral side lobes. The lateral lobes possess muscle bundles, whereas the dorsal and ventral lobes tend to be simply thickened cuticles, except the anal segment. What matters here is how the muscles are inserted in the structures. Herein, I use the term intrinsic and extrinsic muscles, which are distinct from each other in terms of the configurations. Intrinsic muscles shown by orange arrowheads are those with both ends are attached to the structures, which I mean in this case are lobes, whereas extrinsic muscles shown in green arrowheads have one end inserted in the lobes at an oblique angle. The difference is significant from the functional aspect. From left to right, this panel shows the serious sections of the thoracic and abdominal segments of Yogma bird pecten. You can see that the lateral lobes contain intrinsic muscles like that, like these arrowheads, from the very beginning of the thoracic segment to the anal segment. The musculature is particularly relevant to locomotion. The crawling can be explained by three phases of muscle contractions in reference to the well-studied examples in caterpillars. I'm not going to so deep in this direction, and it takes a little bit of time to explain the mechanism. In a nutshell, the segments need to be sequentially lifted up from the substrate when a caterpillar moves forward, as illustrated by Gillett, 1995. In the process, the dorsal longitudinal muscle and those ventral muscles are contracted, and that is followed by the contraction of intrinsic and extrinsic muscles within the proleg. For here, intrinsic muscles and extrinsic muscles are named as retractor muscles and dorsventral muscles, respectively. These extrinsic muscles in the prolegs are particularly important in propelling the larva forward. Based on the similarity in the structure and larval movement, I hypothesized that the intrinsic muscles of lateral lobes and cylindrotomians can be comparable to dorsventral muscles in caterpillars' prolegs. The extrinsic muscles are comparable to the retractor muscle of the proleg. Given that, the lateral lobes can be a functional analog of prolegs in the caterpillar. With this regard to ascertain my hypothesis, I should compare the musculature of cylindrotomians with the other clades of diploids. Here is a summary of the function of the cuticle lobes. First, the gill hypothesis was not supported by my observation because the spiracles of aquatic species are functional. So the cylindrotomians can intake oxygen through the spiracles. Second, internal musculature and its configuration suggest that the lateral lobes can be involved in locomotion. So overall, the elongated cuticular lobes may facilitate the resemblance of cylindrotomians to host plants and simultaneously be used for different purposes, like locomotion. What puzzles me is the evolutionary background of moss mimesis, because knowledge about keen-sighted animals that use moss patches as a hunting ground is lacking. This is a quote by August Thienemann, who was the first person who touched on the evolutionary background of the larval forms of Cylindrotomian. He noted, only birds could come into question. I'm actually sure that those moss cushions in which, e.g., the Diugma larvae are not normally searched by birds. But in any case, on my many excursions at such places, I have not been able to make a single corresponding observation. So the absence of keen-sighted animals like birds is inconsistent with an evolutionary scenario of anti-predator adaptation in such environments. However, many arthropod groups resemble mosses. This fact seems to be a piece of circumstantial evidence for the presence of visual predators on moss patches. So I advanced the research to answer the question Thienemann proposed a century ago. Last autumn, my students and I started recording animal behavior on moss patches to ascertain animals using moss patches as a hunting ground. We set motion-triggered camera traps in this way, directed to the moss patches at the center. We are facing technical problems with this methods, like the effective capture rates are pretty low, and it seems that the cameras sometimes miss the right moment. But I hope our assessment will pay off eventually. The first bird detected is the Eurasian wren, Troglodytes troglodytes. It's one of the smallest bird species in Japan and forms a territory along the stream and constantly forages for insects on moss patches. The cameras have also detected some other birds, like the blue and white flycatcher. I like to show you the notable behavior of undistributed birds. Let me play the video that shows a bird collecting mosses, probably for the nest building behavior. Please pay attention to the middle left. So now she's plucking some mosses from the fallen tree, and now she's slamming it against the tree trunk. She repeats it for several times. So now we have a prospect of revealing how mosses provide ecological niches for forest passerines' foraging sites and nest materials. Both perspectives are essential to understand the ecological functions of mosses. The material compositions often differ between outer and inner layers. The outer layer provides structural rigidity, but the inner layer is chosen for the good of incubating eggs and chicks. So I'm interested in the functions of bryophytes as nesting materials. Actually, the intersect of ornithology and biology is fully studied yet. For example, nests with abundant mosses in the outer layer can ensure a cryptic appearance. In addition, some previous studies show that nests with bryophytes can benefit from adjusting microclimate in the incubating place, like temperature and humidity, and also antibacterial effects against pathogens. So as a final note, I'd like to develop my research further in collaboration with people and gain deeper and wider perspectives to understand the intricate web of biological interactions that structures the forest ecosystems.
This is the end of my talk. It would be great if you learned some new perspectives on the ecological functions of the smallest element in the forest ecosystems. There's a small world in there where the mosses, insects, birds, and other organisms influence each other's evolution. Thank you so much for watching this video.